Hey everybody, welcome to Acid-Base Titration Lecture. We're going to get right into it. I want to show you something here that we have on this, first slide, your introduction slide. But notice where it says strong base, that's your titrant. Your titrant is going to be anything that's actually going to go into your burette, okay? And then, of course, your weak acid. But this is a really fun lab. We're going to get into it. You're going to see some color changes. It's all about precision, accuracy, and you're going to learn that how important accuracy and precision are. But you're also going to learn a lot of very valuable laboratory skills. You're going to be learning about a strong acid, strong base titration curve, and a weak acid, strong base titration curve. This is just something I want to show you. You're going to be learning how to calculate grams that are going to be required to achieve whatever molarity you're actually looking for. You're going to be working with a solution to find that molarity. You're going to figure out how to use mass and volume data to determine the molarity of a solution. You're going to get a chance to make a chemical solution, which is always cool. You're also going to figure out how to do a proper titration to find the molarity of your solution. And then you're going to learn how to use a known solution molarity with volume data to find molarity of a second solution. So just a quick reminder, this is basic dimensional analysis. We're converting moles to grams or grams to moles. If you in the problem that you're working, if you know the number of moles, then you know how to find your mass. You're going to put your grams over your moles. Same difference. If you have your mass and you're trying to find your moles of your solution, you're going to use moles over grams. And you'll take that and go into dimensional analysis. If you're struggling with that, there is a video for you in your video library in D2L. So let's just talk about this one. So your equivalence point for an acid-base neutralization reaction is the amount of base that you add to equal the amount of acid initially present. One more time. It's the amount of base that you add to equal the amount of acid initially present. So what that means is that the acid has been completely neutralized. The point in a titration where the indicator changes color is called the end point, and you're going to get to see that firsthand. It's actually really cool. So an indicator is selected so that the color change is going to occur as close to the equivalence point as possible. For any given acid-base titration, a good indicator changes color over a pH range that coincides with the portion of the curve near the equivalence point where the pH rises really sharply. Okay, it rises really sharply. Let me show you. The pH at half equivalence can be used to identify the unknown weak acid. Okay, the equivalence point occurs at the midpoint of the region where the pH rises really, really fast, and you can see that on this graph. This is going to be your setup for the most part. In a titration, the titrant is added from a burette, and I wanted to point that out from you before. It is your titrant that is inside your burette. Now, maybe in some other experiment, not this one, but you have water in there, then water is your titrant, okay? So the titrant is added from your burette into a flask with a measured amount of another substance in a solution, okay? So you're going to measure some solution and put it into your flask before you start to titrate until the reaction between the two is complete. Okay, so once you will see when it is complete. I don't think you should worry about that at all. So now, because acidic and basic solutions are generally clear and colorless, you have to monitor the progress of this reaction very closely, and you're going to use an acid-base indicator or some other substance that changes color with the pH. So most commonly used indicators are methyl red, bromethymol blue, and phenolphthalein. So phenolphthalein indicator is going to be in pink form. And so this is kind of what we're working with on the pH scale. So these indicators change color between your pH 5 to pH 9 because the complete neutralization of most of your acid and bases generally occur within that pH between 5 and 9. Okay, so an acid-base indicator changing color indicates the reaction between the acid and the base is complete, and this is the actual endpoint of your reaction. Okay, it's the actual endpoint of your reaction. So phenolphthalein is colorless below the pH of an 8.0. Okay, it gives a sharp endpoint, meaning that when a titration is being carried out, the phenolphthalein is so sensitive that within a fraction of a drop of a titrant, it's going to make the solution turn completely pink. Okay, and it turns completely pink when the solution is basic. So that's what we're working with. Phenolphthalein is colorless below the pH of an 8.0, right? But it turns pink when the pH is 8.0 or higher. Just to throw that in there for you. That way you have an idea going into it what we're talking about. So the fainter the pink color I was just talking about, the closer to the equivalence point. The thing about it is it does happen very quickly. You're going to want to watch it fairly closely, very closely, I should say, and you'll be swirling your flask. It's going to look a lot like this, what you see on the screen. And just, you know, your first one might be a little bit dark, and then when you do your second trial, try to get a little bit lighter. Third, try to get a little bit lighter, and it's all about precision and accuracy. So let me just state, the terms endpoint and equivalence point are not synonymous. They do not mean the same thing. Your endpoint is the moment in the experiment where the indicator changes color. Okay, the equivalence point is just a condition. It's theoretical, and it's a condition when the titrant has completely neutralized. So they are not synonymous. They do mean different things. In an acid-base titration, the changes, the reaction completion point, the moles of acid titrated, they equal the moles of base that's used. Okay. Burettes. Now, we've talked about burettes a little bit. They're used to deliver a recorded amount of liquid to another container. It's going to be marked in milliliters, just like the graduated cylinder on your right, but it shows 0 mils at the top, and the numbers increase as you go down. Remember the stopcock. It's your little handle. It's what's stopping the liquid from dropping through. It's right there on the right on the burette on the picture. Your stopcock controls the liquid flow when it's open, when it's parallel. So if you look at this picture, this stopcock is open. So as you would know that it's going to be dripping when it's going to be open. Okay, let me just show you. This is just like the one you're going to actually see during class. You're going to have to wash and rinse your burette, okay? What's going to happen is you're just going to rinse it with water, okay? We're rinsing with DI water. You're going to go over to the sink and open your stopcock, and you're going to let it drain out the tip. You're actually washing the tip at this point, okay? At that point, you're going to close the stopcock, and we're going to condition the burette. Basically, what we're going to do is after you release all that water, and you can do that in the sink, you're going to close the stopcock, and you're just going to put about 5 or 10 milliliters of the titrant, which is what's going to be used through the burette. You're going to put just 5 to 10 milliliters in there. You don't have to measure it. Just put some in there. You're using it to condition. You're never going to be able to read 5 or 10 mils on a burette anyway, so don't try to break your back doing that. So you're going to tilt it sideways, and you're going to roll the barrel to completely rinse the inner walls, and I'll show you how to do this in lab. You want to make sure that the tip is also conditioned. So you're going to actually go into a waste container, because we never put chemicals down a drain. You're going to go into a waste container, and you're going to open that stopcock over the waste container, and you're going to let it drip through, and that conditions the tip. You'll do that 2 times. All right, you'll do that 2 times. So then once you're done, basically, you're going to have to fill the burette. You're going to close the stopcock and use a clean funnel. You'll probably find one in your tray or your drawer. And you're going to fill the burette with titrant just above the 0. You just place a container under the burette and open the stopcock briefly to fill the burette tip with solution, making sure that there aren't any air bubbles in it, and to get that level of meniscus to fall within the marking of the burette. Make sure the stopcock is in the closed position or you're going to lose your titrant, okay? Your initial level of titrant doesn't need to be exactly 0. And the reason why is because the initial level is going to end up being subtracted from the final volume to determine how much you actually delivered, okay, out of the burette. Now, for any measurements, when we're recording record the volume of the titrant by reading the bottom of the meniscus, okay? You're always going to refill the burette before each trial to ensure that there's enough titrant that you're going to be able to get through all of your experiment, all of your trials, okay? Sorry about that. Anyway, so we're talking about reading the meniscus. When we're reading the meniscus, you're going to read the very bottom. And we've drawn one here for you with the black line. So you see the very bottom of your curve, which is right here. The very bottom of your curve, that's where you're reading it. So if you would read this one, where is 25? This is 25 right here, right? And it falls just below 25. So you might call that 29.8, 29.7, anything in that ballpark, that's going to be a valid answer, okay? But back to the burette. Once we're done, we've read our burette, we're getting ready to clean our burette, you're going to empty the burette into your waste container that you have been using the whole time. You'll rinse it out with water, and you can use tap water for that, but that waste also needs to go into a waste container. Then you're going to rinse it. Make sure you're running, when you're rinsing with any kind of water, tap water or DI, you're letting it go through the tip also, so you're washing the tip out also. Then you'll do one final rinse with DI water, and you can actually let that go into the sink. Don't get confused. Just use your waste container if you don't want to remember. But just do a final rinse with DI water, leave the tip open, return it back up to the teacher's desk or the cart at the front of the desk. All right. So we're also going to use volumetric pipettes. Now, volumetric pipettes, they deliver a specific amount of liquid to go to a degree of accuracy. So you're going to rinse the pipette the exact same way. Place the pump on the top, roll it up, pull the solution up, and discard it into a waste container. Again, I will show you how to do that while we're in class. And this is basically what we're looking at. We're going to condition the pipette. You're going to draw it up with a roller. You're going to put it in a waste container, not into the beaker that you're going to dilute your reagent. You'll repeat that process 2 times. And this is what it actually looks like. And I just wanted to show you, find your calibration line. You're not going to see it like this on yours. Just take your fingernail and run your fingernail across and you'll feel it. Okay. Your solution goes up to that. That is your 10 mil mark on this particular one. So if you're using a 1 mil, that's your 1 mil mark. If you're using a 2 mil, 2 mil mark. 10 mil, 10 mil mark, okay? You're going to press this pump's release button to release the solution. And again, I'll show you how to do that in class. Just a quick reminder about moles to grams, grams to moles. You're going to figure that out with how much NaOH that you need, okay? So be ready for that. These are available to you in your lab, this slide, and this slide. They are all available to you in your lab. I need you to look over those. If you have questions, please bring them to class with you so we can address those questions in a readily manner. So that's it. That is it for our titration lecture. If you have questions, make sure you write them down or put them in your lab notebook, and I can certainly respond to them via your lab notebook also. So good luck in titration and have fun with the color changes.